Hello lovelies, in this video we're going through the foundation content for your OCR Gateway Biology paper 2. Now, this is a really low-key, chilled, stress-free way for you to revise. If you want to, you can make some notes, or you can use the free questions and the flashcards over on the website. While you're there, you can also have a look at the predicted papers and the walkthroughs that we've done for the predicted papers.
The carbon cycle. All materials in ecosystems are recycled to provide the raw materials for new organisms to grow. The carbon cycle returns carbon from carbohydrates, lipids, and proteins in organisms to the atmosphere as carbon dioxide, so that plants can use it for photosynthesis to synthesize new molecules like glucose. Decomposers break down dead organisms and return nutrients to the soil for plants to take up through their roots. Photosynthesis is the only process to remove carbon dioxide from the atmosphere. Decomposers, bacteria and fungi release carbon dioxide as well when they respire during decay or decomposition. Fossil fuels and the production of fossil fuels like coal and oil takes millions of years to form, and this is trapped carbon from dead organisms that hasn't decayed. Wood burning and burning of fossil fuels for transport and in factories in order to produce energy, these are all human activities, and these are what put carbon dioxide into the atmosphere, but these are processes that humans have caused that have caused the carbon cycle to become unbalanced. The water cycle is important as it provides fresh water on land for plants and animals before it returns to the sea. The first stage is evaporation, where water evaporates from large bodies like the ocean due to heat energy from the sun. Then it condenses, so it cools down to form clouds. Water also evaporates from leaves through transpiration. Precipitation falls from clouds either as rain, sleet, or snow. And then the water percolates through rocks into the ground and runs off of rivers and ponds and other things like farmland back into the ocean. In the water cycle, water is continuously evaporated and precipitated. We need to know the organisation of an ecosystem. Feeding relationships in an ecosystem can be shown in food chains. Food chains always start with a producer. These are always photosynthetic organisms, as these are the source of all biomass on Earth, because they can use light energy to synthesize molecules like glucose. The arrows in a food chain or food web show the transfer of biomass, and by biomass we mean living tissue that is made up of the molecules carbohydrates, lipids. Primary consumers are the first organisms that eat another organism in the food chain. They can often be herbivores, which means that they only eat plants, and they will be prey species because they will be consumed by other animals. The secondary consumer is the second organism to eat another organism in the food chain. They are predators because they catch and consume other organisms, but they can also be prey to organisms higher up the food chain. Tertiary consumers, and then we also have quaternary consumers as well. These are predators and they're often carnivores, so they only eat animals. The top predator in the food chain will be the apex predator, and they will have no predators that eat them. Abiotic factors are non-living factors that can affect the number of species in populations in an ecosystem. They can also lead to species having specific adaptations to survive in extreme habitats. Light intensity is an abiotic factor. It obviously affects photosynthesis and can cause sunburn in animals. Temperature affects all organisms because it affects enzymes. It also has an effect on the rate of transpiration. Wind intensity or direction, again, affects transpiration. could potentially affect flight, and it can cause exposure issues in animals. Moisture levels or water availability. All organisms need water in order to survive. Soil pH and mineral ion concentration. Again, pH can affect enzymes of organisms living in soil, and plants need mineral ions in order to survive. The concentration of oxygen is an abiotic factor in water for aquatic animals. And the concentration of carbon dioxide is obviously a factor, but only for plants because it can affect the rate of photosynthesis. This is mostly not an issue unless these plants are in some kind of greenhouse or container. Biotic factors are caused by living organisms that can affect the size of populations in an ecosystem. New predators. Prey species may not be prepared or able to adapt to avoid predation. New pathogens. Organisms will have no resistance to the disease if they've not been infected with it before. Amount of food. Limited food due to a reduction in population size can increase competition and reduce survival. Equally, when there's lots of food, population sizes can increase. Competition. Competition with other species for resources could lead to one outcompeting the other until there are not enough individuals of that species left to breed. This means that they could go extinct. An example I've got here is the red squirrel and the grey squirrel. There are very few red squirrels left because the grey squirrel is able to outcompete them in most habitats. We need to be able to explain how biotic factors can affect population size. An example with disease. What would happen if a new pathogen reduced the number of birds in this community? It would increase the numbers of snails and worms, as they now had fewer predators. But the number of lizards and snakes could decrease, as the eagle now has to eat more lizards and snakes to make up for the lack of birds, as they are one of their food sources. So when we look at food chains like this and talk about the impact of a biotic factor on a community, we have to look both above and below in the food chain. A food example, we need to know about predator and prey population sizes, how they're linked, and how they often cycle over time in stable ecosystems. There are certain rules that these predator-prey cycles always follow. As prey numbers increase, the predator numbers will increase because there's more food, so more of them are able to survive and reproduce, but there will be a slight delay. If there are too many predators, or as the population size of the predators increases, they might eat too many prey, and so then the predator population size will decrease because they now have a lack of food. When the predator numbers are low or have decreased, fewer predators means that the prey numbers can then increase because more of them can survive to reproduce. If you were sketching these graphs, remember there can never be more predators than there are prey. And also the predator graph should always be lower and shifted to the right. In an ecosystem, there are individual organisms, populations, which are a group of individuals of the same species, communities, which are all the populations of all the different species in an area, and the habitat, which is the non-living parts of the environment. An ecosystem is the interactions between the living and non-living parts of an area, so all of the communities in a habitat. Interdependence describes how species in a community depend on each other. They can depend on each other for shelter, pollination, seed dispersal, and food. If one species is removed, it can affect the size of other populations in the community. Stable ecosystems have balanced environmental conditions, and the species numbers in the populations are relatively constant. Stable ecosystems take time to develop, and so examples are often hundreds of years old, like large coral reefs or ancient woodland. Keystone species are species that, if they're removed or lost, will have an incredibly great impact on the ecosystem. Competition. All organisms will compete with other organisms for resources in their environment. So for animals, they compete with each other for food, space or territory, and mates. And plants compete with each other for light, space, water, and mineral ions. Competition can either be between members of the same species, we call this intraspecific, or between members of different species, this is known as interspecific. Competition leads to adaptations that allow organisms to outcompete others in their habitat in order to survive. We're going to look at adaptations in more detail, but examples of things like storing food that squirrels do to make sure others can't have it and make sure they have a store for themselves, long roots of plants in the desert so they can get more water from other plants, and then things like horns or claws, adaptations for fighting that allow them to fight for other mates or other things like food. Parasitism and mutualism. Parasites live in or on other organisms called hosts. Parasite takes nutrients from the host and it receives nothing in return. So this means there's no benefit to the host organism of having the parasite, and often it can be detrimental to the host organism. An example is fleas on cats and also on dogs and other animals. Fleas feed on the blood of the cat. They also live on the cat, which provides it a habitat as they are attached to their hairs, and they can jump large distances, which allow them to jump from one organism to another. Many parasites also lay large amounts of eggs to try and increase their chances of reproduction, just to make sure that their offspring have a chance of some of them surviving and finding a new host. Parasites often have special adaptations to take as much as possible from the host, but without killing it. Another example is a tapeworm. A mutualistic relationship is where both species benefit. An example is bees on flowers. Bees get nectar from flowers, which is their food, and the flowers get pollinated, so they get help with their sexual reproduction. Lichens are another example of mutualism. They are actually two organisms, an algae and a fungus, and these actually live sort of together in this symbiotic relationship. The algae photosynthesize and therefore produce sugars which they share with the fungus, so they provide it with food, and the fungi provides shelter for the algae from harsh climate. In most populations, there is variation, which just means differences in characteristics between organisms. Genetic variation is caused by mutation. Most of the time, this has no effect on the phenotype, but sometimes it can change it. For example, a new allele for fur colour in my cats could have been caused by a mutation. Variation can be the result of inherited genes, the environment, or a combination of both. Some examples of different characteristics caused by inherited genes are eye colour, blood type, and earlobe shape. Some examples of varied characteristics caused by the environment only are scars, the language you speak, and your hair length if you get it cut. And there are 2 or 3 characteristics that we can talk about being affected by both genetics and the environment. So height and body mass, although there is some element of genetic effect on these in your characteristics, both can also be affected by things like diet, nutrition, and exercise, or things like light intensity or mineral availability if it's a plant. And then flexibility is another example. So yes, genetically you could be more flexible than someone else, but there is also the ability to improve your flexibility through practice. There are 2 forms of variation. The first is continuous variation. It's often shown as a histogram like this, where we've got multiple bars touching, and they normally make this kind of bell curve shape. It's when there is a range between 2 extremes, and most individuals are close to the mean. So this will be the peak in the middle. These are often controlled by more than one gene, and they can also be influenced by the environment. Examples include height, leaf length, or the number of flagella on a bacteria. The other type of variation is discontinuous variation. This is where we can see another bar chart or histogram, but these bars are separate from each other in separate categories. It's when there are 2 or more distinct categories with no values in between. Usually regulated by a single gene and not influenced by the environment. Examples include blood group, flower colour, or sex. So you can only have one blood type, you can only have a specific flower colour, and there will be potentially different ones, like different blood types, but you can only be in one group at a time. These show that some characteristics that show discontinuous variation are controlled by single genes, but most are controlled by the interactions of multiple genes together, which is what creates the continuous variation. Sexual reproduction involves the fusion of male and female gametes that are produced by meiosis. So we have eggs and sperm in humans, as the female gamete is an egg and the male gamete is the sperm. In a plant, the female gamete is also an egg and is also produced by meiosis in the ovaries, the same as in humans. And then the male gamete is pollen, and this is produced by meiosis in the anthers. The male gamete of sperm in humans is produced in the testes. Sexual reproduction mixes different DNA from each parent, creating variation in their offspring. Gametes are non-identical, and the fusion of each pair of gametes, so which sperm and which egg meet each time they fuse, is random. So this helps to shuffle and mix the DNA to make sure that every offspring is genetically unique. Asexual reproduction is carried out by all bacteria, some plants, and a few animals. This example diagram is bacteria dividing by binary fission, which is very similar to mitosis. No gametes are involved in asexual reproduction. There is only one parent cell, not 2. There is no mixing of DNA, so there is no genetic variation in the offspring. They are all identical. It relies on mitosis instead of meiosis to produce genetically identical clones of the parent. Meiosis is the type of cell division that produces gametes, or sex cells. It occurs in reproductive organs, for example, testes and ovaries in humans. To start with, we have a normal body cell that is diploid, or has the full number of chromosomes that a normal body cell would have. In this example, you can see 4 chromosomes. Then the DNA replicates or doubles or copies itself to prepare for cell division. So now we have 8 chromosomes. At this point, this is exactly the same as the start of mitosis. Then we have our first cell division where that cell splits into 2, So each cell now has 4 chromosomes again. Then each of those cells splits into 2 again. So we have our second cell division, and this produces 4 non-identical cells with half the number of chromosomes, in this case 2, of the original parent cell. We call these cells haploid. So diploid cells have the full number of chromosomes of every cell in the body, and haploid have half the number of those chromosomes. The reason these 4 cells are non-identical is that during meiosis, the DNA is shuffled in order to make sure that each of these gametes will be unique. We need to be able to compare meiosis and mitosis. So unlike mitosis, there are 2 divisions, not 1. There are 4 cells produced, not 2. And the 4 cells produced are non-identical and they are haploid, or contain half the number of chromosomes. Let's look at what happens and why meiosis is important producing these gametes once they are ready to be used in sexual reproduction. So now we are looking at 2 human gametes, an egg cell, the female gamete with 23 chromosomes, which is half the number of the normal body chromosome number in humans, and sperm cells have 23 chromosomes as well in their nucleus. During fertilization, the nuclei of each gamete fuses and the full number of chromosomes is then restored to form a diploid zygote, which is the new cell that's going to form the new organism, and that will now have all the 46 chromosomes it needs in order to carry on developing and growing into a human. That single cell will start to divide by mitosis to form identical copies of itself to create an embryo, which is a ball of stem cells. And then those stem cells keep dividing by mitosis to increase the cell number, but they also go through the process of differentiation to produce all the different specialized cells needed to make a fetus. It's important that we understand the role of meiosis and mitosis. So we need meiosis to make the gametes that have half the number of chromosomes so that the DNA can be mixed when the 2 fuse together during fertilization to create a cell that has the full number of chromosomes we need for a body cell. And then we need mitosis to multiply that cell number, making identical copies in order for a fetus to grow. And we need those cells to go through differentiation to become specialized to form all the specialized cells and tissues in the body. A gene is a short section of DNA that codes for a protein. An allele is a different version of a gene that gives a certain characteristic. The genotype is the alleles an organism has for a particular gene. The phenotype is the observable characteristics that an organism has based on their genotype. There are 3 main genotypes. Homozygous recessive. This means you have 2 lowercase letters to represent your alleles because both alleles in this genotype are recessive. Recessive alleles need 2 copies in order to be expressed in the phenotype. So for example here, the phenotype will be white flowers because both of these alleles are recessive and they both code for white petals. Homozygous dominant is a genotype where both alleles are dominant alleles. Dominant alleles are presented in the phenotype when there is only 1 copy of them. So you only need 1 allele to be dominant in the genotype for that phenotype to be expressed. Then there are heterozygous genotypes. This is where we have 1 dominant and 1 recessive allele, or 2 different alleles. Both, in this case, the heterozygous and the homozygous dominant genotype will produce the same phenotype, which is red flowers in this case, because both of these genotypes have at least 1 dominant allele for red flowers. We can use Punnett squares to predict the possible genotypes and phenotypes of the offspring of 2 parents. So let's look at an example where we have crossed two heterozygous red flower plants. So they're both heterozygous, so their alleles need to be one dominant and one recessive. And then we put these into the Punnett square to show what the gamete alleles could be. So each plant could either give a dominant allele big R or a recessive allele little r into their gametes. And remember, the gametes would be the eggs and the pollen in this case, as they are plants. We then fill in the Punnett square by going down and across for each box to fill in from the gamete what they would receive from the parent and put them together. So this gives us our possible offspring genotypes. So we have one homozygous dominant with two capital R's, two heterozygous offspring with one capital R and one lowercase r, and one homozygous recessive offspring with two lowercase r's. Then we can work out the phenotypes. So I've put the red and white flowers on to show you, but obviously anything that has at least one capital letter or one dominant allele will have a red flower phenotype, and then the two recessive alleles for the little r's, they will have the white flower phenotype. We can either use ratios to describe the phenotypes, so for example 3:1 red to white, or we could use percentages to look at the probability of the offspring being a certain genotype. For example, we can say there's 25% chance that the offspring will be homozygous recessive. When doing this in the exam question, you should always annotate the Punnett square to show what you mean. So if I'm saying there's 25% chance of being homozygous recessive, I circle that offspring genotype on my Punnett square. In humans, there are 23 pairs of chromosomes. 22 are body chromosomes, and pair 23 are the sex chromosomes. You either have XY or XX, and these sex chromosomes play a role in determining sexual characteristics. Every time a sperm and an egg fuse in fertilization, there is a 50% chance that the embryo made will be either male or female. We can show this with a Punnett square. Egg alleles can only be XX, and sperm alleles could either be X or Y. There is 2 out of 4 or 50% chance of getting an XX combination, which will produce a female, and there is 2 out of 4 or 50% chance of getting an XY combination of chromosomes, which will be a male. Classification groups organisms depending on their structure or characteristics. This system was developed by Carl Linnaeus. Here we have an example of the classification of a lion. We need to know the groups in order, so kingdom, phylum, class, order, family, genus, and species. And as we go down these groups, we get more and more specific to the characteristics that are more similar to the lion. So you need to be able to remember these in order, so there's a way to remember them that I've put here. King prawn curry or fat grated sausages. That can help you remember the first letters in the right order for all of those words, but you will need to be able to know that in order. What you don't need to know is the other side, which is the actual Latin names that the groups are for the lion, for example, but you'll be shown examples like this in the exam and need to fill in the gaps or be able to say what the phylum is for the animal when you're given this information, for example. Here is the binomial species name for the lion, which is Panthera leo. It uses 2 words, so it uses the genus and the species name to build the binomial name. This is really important because it means that it's a universal name and language that can be used all over the world instead of the common name like lion, which would change from country to country. It also helps having the genus as part of the binomial name so that you can use it to see evolutionary relationships, to see what other animals will be in the same genus and therefore will be closely related to the lion. We also need to know the definition of a species. A species is a group of similar organisms that can breed to produce fertile offspring. Originally, kingdom was the highest group in this organisational system with 5 different categories: animals, plants, fungi, protists and prokaryotes. This was the system developed by Carl Linnaeus. The development of electron microscopes and understanding of biochemistry caused a new scientist called Carl Woese to update the classification system. This is known as the three-domain system. So a group above kingdom was added called the domain, and there are three domains: archaea, bacteria, and eukaryotes. And then each of these has kingdoms within them, so it's just the kingdom archaea, the kingdom bacteria, and then the eukaryotes has all of the other eukaryotic kingdoms. It is just the prokaryotes that have been split into these two new groups, archaea and bacteria. So the kingdoms in the eukaryotic domain are plants, animals, fungi, and protists. These were split because it was found that these two groups, archaea and bacteria, within the prokaryotes were significantly different enough from each other to be two separate groups. So the archaea are ancient or primitive bacteria and mostly extremophiles. And then the bacteria are the true bacteria, what we would know as something like E. coli that we can work on in the lab. And then the eukaryotes are grouped together because all of their cells contain a nucleus. Evolutionary trees. By comparing DNA sequences between organisms and using fossil evidence, we can show how closely related different organisms are. This information can then be used to build evolutionary trees, which shows how organisms evolved through time. So here you can see we have a single shared common ancestor for all of the species A, B, C, D, and E in this group. Then we have a branch point, and A branches off first. So they were the ones to evolve first, and therefore they're the oldest species. Then when we have a second branch point, B and C split in one direction, and D and E split in another direction. And this is because D and E share a common ancestor between them, and B and C share a different common ancestor between them. D and E are more closely related to each other than any other letter here, and B and C are more closely related to each other than any other letter here because they share a branch point. And B and C actually split or evolved more recently because you can see their branch point shows a more recent common ancestor in time if we're using the kind of y-axis of time on the left. Previous classifications based on observable characteristics alone was known as artificial classification. This can now be updated by comparing DNA sequences to see how closely related organisms actually are, and now this is known as natural classification. Evolution is the change in inherited characteristics of a population over time. It happens through the process of natural selection. Natural selection is where only some organisms with certain characteristics can survive in order to reproduce. Over time, this makes them well adapted to survive in their environment. And here you can see the tree of life, this idea that shows that all of these species are connected to a single living common ancestor from billions of years ago. We need to be able to describe the process of natural selection. There's variation in the population due to random mutation. In my example here, I've got a population with 3 different phenotypes, A, B, and C, which would have been caused by mutation. Some organisms have characteristics that enable them to survive and reproduce. So we've got 2 changes that happen over time, and in each one of those, one of these phenotypes does not survive, and the others go on to be able to survive and therefore reproduce. They pass on alleles for these characteristics to their offspring, so then there are more of those alleles and more of that phenotype in the population. So you can see how over time, the amount of phenotype C in the population increases until that's the only phenotype. So over time, this leads to populations having phenotypes that are best adapted to their environment. If this happens in 2 populations of the same species, they can become so different and adapted to different environmental conditions that their phenotypes mean they're so different they can no longer breed together to produce fertile offspring, which means they produced 2 new species. This last point is an add-on, but for the first 4 bullet points, these are the exact language and the exact wording we should use when describing the process of evolution by natural selection in the exam. Fossils are the remains of organisms from millions of years ago, which are found in rocks. We can use them to show how much or little different organisms have changed over time. There are 3 different ways that fossils can be made. When parts of organisms that didn't decay due to the lack of conditions, so either lack of oxygen or certain temperatures, they can be fixed in time, either in amber or ice. Soft parts of the animals can decay, but the hard parts are replaced by minerals. This is known as mineralization, often after being buried in sediment. Traces or imprints of organisms such as footprints or their burrows can be preserved in rocks. We need to be able to explain why the fossil record is incomplete. Many early life forms were soft-bodied, so all of the parts decayed. This is the reason that scientists cannot be certain how life started on Earth. Many fossils have been destroyed by geological activity. Imprints can be easily washed away before they have a chance to become a fossil. Many fossils are still undiscovered. We do not know how many there are still buried. And not all organisms become fossils because the conditions to make them are very rare. Antibiotic resistance. Some bacteria in a population have the ability to survive treatment with an antibiotic. These will survive and reproduce, passing on the genes that allow them to survive to their offspring. Over time, we get an increase in frequency of the resistant bacteria in a population until we have a whole population of bacteria that cannot be killed by that antibiotic. This is an example of natural selection and therefore evolution of bacteria. An example we need to know is MRSA. We need to be able to suggest ways to reduce antibiotic resistance. We can reduce prescriptions of antibiotics for viral infections, so prevent doctors from giving out antibiotics unnecessarily. Making sure that patients take the full course of antibiotics every time, so they take all of the antibiotics they're given, even if they start to feel better. And reduce the use of antibiotics in farm animals. The development of new antibiotics is expensive and slow, and cannot keep up with the rate of new strains. This is a topic that came up in paper 1, and it can also come up on paper 2. So it's good to know and have a recap of this here because we need to know it for both papers. There are 2 bacterial disease examples we need to know. The first is gonorrhea, which is a sexually transmitted infection, so we can prevent the spread with condoms, similar to HIV. The symptoms caused are discharge from the genitals and pain when urinating, and it can also cause a fever. Salmonella is the second example. This causes food poisoning that we have already mentioned, as it's spread from eating contaminated food, for example, raw chicken. The symptoms are sickness, fever, and diarrhea. We can prevent the spread by washing our hands before and after touching raw meat, and also cooking food to high enough temperatures to kill the bacteria. In the UK, chickens are also vaccinated against salmonella to try and reduce the spread and make sure it's not present in eggs as well. In both of these examples, the symptoms are caused by toxins released from the bacteria. Treatment for any bacterial infection involves antibiotics. These are drugs that kill bacteria in the body or stop them from replicating. Not all antibiotics will be able to kill all bacteria. We now have an example that there are many resistant strains of gonorrhea in the UK specifically, which cannot be treated with most antibiotics. It's not possible to count all the organisms in a habitat, so sampling is used to estimate population size in an area. We normally use quadrats. These are a metal wire frame and there'll be a set size, so for example 1 metre squared, and you would count the number of organisms of each species inside the frame. Random sampling methods are used to estimate the number of a species in a certain area. This is also known as the abundance. As you can see in the diagram, we would set up 2 tape measures in a way of gridding the area, and then we would use a random number generator to select coordinates for the x and y axes in order to say where to place the quadrat. This ensures that the quadrats are placed truly randomly and therefore removes bias. Also, we should place a large number of quadrats. This is to improve our reliability of our experiment. Systematic sampling with a transect is used to see how a changing abiotic factor affects the number or distribution of a species, so where a species is found. So, as you can see from the diagram, we've laid a transect going away from the base of a tree, and we've done it twice. So you would lay a transect along a changing environment, so a tape measure, for example, along a changing environment, and then place your quadrats at regular intervals. That means they should be the same distance apart. So here we've gone for 0, 5 and 10 metres. So every 5 metres along my transect. You should repeat the whole transect in a different position. So as you can see here, we've got 2 of them. The change in the environment should be the same. So as we said, moving away from the tree and starting from the base of the same tree. But there should be no overlap of your quadrats, so you're not sampling the same area. For both, you would calculate a mean across your quadrats. For random sampling, you could then multiply it by the area of the field that you are measuring to estimate the total population size. For the transect, you would take the mean from each of the quadrats from the same distance along the transect to show the mean number of each species at each distance. For sampling animals, different techniques and equipment must be used because animals are able to move around, unlike plants. So some of the examples of techniques or equipment you can use to catch and therefore count or sample animals are pitfall traps. So this is where there would be you dig a hole and there would be cover it with leaves and things, but the insects would be able to walk across and then they would fall in and be stuck. So you can observe them, count them, and then obviously you would release them. This is suitable for ground insects and small animals. Sweep nets, these are big, long nets that you can use to either catch flying insects or in aquatic systems like in rivers or ponds to be able to catch organisms in the water. Hooters allow you to suck up, so literally use a little bit of gentle suction to suck up specific insects into the little pot. You can then obviously observe them, check what species they are, and then you can obviously release them. This is for very small insects like ants and things like that. We use something known as the capture-recapture method in order to estimate the population size of animals in an area. So first of all, we need to trap or catch the organisms using one of the methods we've already talked about, and then we need to mark them in a harmless way. This could be putting like a dot of nail varnish on them or something non-toxic, or a tag that is easily kind of identifiable but isn't something that's going to harm them or cause them to be more likely to be predated upon, for example. Then you would release them back and then return a few days later and reset the same traps in the same places. Then you would count the number of marked and unmarked organisms caught in the trap. We can use the following formula to calculate population size. So this would be the number of organisms that you trapped in the first place, that you caught in the first trap, multiplied by the number in your second trap, and divide that by the number of marked organisms there are in the second trap. This method relies on some assumptions. First, that there are no deaths, so none of the animals you trapped in the first place died, or none of those animals that you trapped in the first place have left the area. It relies on you doing an identical sampling method each time, so that should be the same traps in the same places at the same time of day, and ideally a similar time of year. It also should be that the marking has no effect on the organism's survival, so it doesn't increase their chance of being predated upon, and it means that they can have gone about their natural business. It doesn't change their lifestyle or their chance of being caught by a predator or their ability to feed or anything like that, which could have affected their chance of surviving between the two trap times. There has been a massive population increase in humans because we have better medical facilities and better access to food, so people are living longer. This means because there are more people, we need more space to live and also to grow food. And we also have increased living standards, so we want more space and more food. This creates pollution from mining, building, energy production, farming, and that all damages natural habitats on land and in water. And because there's more people and we have more stuff, we are producing more waste. All of this pollution reduces biodiversity because it kills plants and animals. Land pollution, which comes from filling up landfill with waste, but also toxic chemicals in the soil, which can come from things like pesticides and herbicides being sprayed onto the land. Water pollution comes from sewage or fertilisers from farmland, and both of these create a low oxygen environment if they run into water, which is done through eutrophication, and it causes the death of animals like fish in the water. Or it could be toxic chemicals from mining or from nuclear power stations. Air pollution is caused by smoke and gases that have come from burning fossil fuels. So the gases include carbon dioxide, but also sulfur dioxide. The smoke and actually breathing in the particulates can harm lungs of humans and other organisms living around towns and cities. And the gases, including the sulfur dioxide, cause acid rain, which damage trees, plants, and cause acidification of lakes and ponds. The greenhouse gases, including carbon dioxide produced, also contribute to global warming. Deforestation occurs to clear land for building and farming. Chopping down trees reduces biodiversity because habitat and food sources for animals are removed, and the trees and plants are killed when they are cut down. Another disadvantage of deforestation is that it reduces the carbon dioxide that's removed from the atmosphere through photosynthesis, as there are fewer trees. This contributes to global warming. The burning of wood or leaving wood to decay that's been chopped down also releases carbon dioxide. And removal of trees increases soil erosion and flooding. When we say that land is cleared for building or farming, in terms of farming, we mean to grow crops for biofuels, or to create flat land to farm cattle, or to grow rice. The farming of cattle and rice both produce methane, which is a greenhouse gas that also contributes to global warming. Global warming is caused by greenhouse gases, carbon dioxide, methane, and water vapour, increasing the atmosphere and trapping more heat energy from the sun. We have to be able to describe how this increase in temperature and climate changes caused by global warming can reduce biodiversity. Increasing ocean temperatures causes carbon dioxide to dissolve in the oceans, which causes coral bleaching or death of the corals. This is obviously a really biodiverse habitat, so this reduces biodiversity in coral reefs. Increasing temperatures causes sea ice to melt, and this reduces ice habitats used by organisms such as polar bears, which contributes to their decline. It also then causes sea levels to rise, which reduces coastal habitats because they flood. Global warming causes lots of weather changes. It can increase flooding in some areas. It can increase drought in some areas. It can increase extreme weather events like hurricanes, tornadoes, and cyclones. This can all cause the change of distribution of organisms, so they move and they might not be able to live where they were living previously, so they often have to move away, and that reduces biodiversity in that area. The changes in climate also affect migration patterns. So there are organisms which migrate long distances from one side of the world or from one hemisphere to another, and changing in the climate might mean that where they were going to or from is no longer suitable for them to live. And so where they are flying to and where they migrate to is going to have to change. Endangered species are species that are at risk of becoming extinct. A species is extinct when there are no individuals of that species left alive. Here's an example from the human evolutionary tree. Only chimpanzees and humans are left alive in terms of species on this tree. All of the other humanoid species that are part of our evolutionary history have gone extinct. Many causes of extinction or causes of organisms becoming endangered are the result of human activity, including things like habitat destruction, hunting or poaching, new competitors. This could be humans or it could be introduced species that humans have brought to the area, and also changes to the environment. That includes things like climate change. In these cases, extinction is often caused through natural selection, where species are less well adapted to cope with these changes, or these changes happen too rapidly for natural selection to occur. Catastrophic events, like a volcanic eruption or an asteroid hitting the Earth, can also cause extinctions. An example was the asteroid that killed the dinosaurs. They are usually responsible for mass extinctions, where many species on the planet go extinct all at once. Humans are continuing to cause extinctions now due to competing with organisms for space, food, and water. So we're destroying their habitats or removing their ability to find food. And because we are changing their habitats too quickly for them to adapt, they are going extinct. Conservation aims to try and prevent species from going extinct. It also tries to help protect habitats and stop them being destroyed. There are many benefits to conservation, such as maintaining biodiversity, ecotourism, which in some places brings in large amounts of money to the economy, knowledge and discovery of new things, like new species that we haven't discovered yet, and these obviously have potential benefits, like being sources of medicines. So there are plenty of medicines that actually started off as plant material or sources that were from microorganisms, and so being able to maintain biodiversity and conserve it so that we can then use these as a potential future source of resources is really important. There are some challenges with conservation. Monitoring large areas is really hard to make sure that all rules are being followed by everybody. Satellites can often be used to help with this, but these are inexpensive. So this is another issue with conservation, is that running the programs are very expensive. Often they're being run by charities which need to raise funds in order to be able to continue their work. Changing laws and legislation to try and protect habitats or species takes a lot of time and often involves many countries having to work together and agree, and often also have funding to make all of these things happen. Sometimes this process can take too long and isn't done in time to save some species. In order for everyone to understand the importance of conservation and be behind conservation efforts, often a lot of education is needed to help improve people's understanding of why we are protecting and how we can protect various species or habitats, especially in their local area. Scientists and conservationists have put programs into place to try and reduce human impacts on biodiversity and help to maintain it. Breeding programs for endangered species to try and increase their numbers. This often happens in captivity in places like zoos. Protection and regeneration of rare habitats to try and make sure that they are no longer damaged anymore and potentially help them to recover. Reintroduction of hedges and wildlife corridors around farmland. On farms, normally only one crop or one species of plant is grown in large areas, so allowing these extra plants and trees and everything around the edges of the field can give homes and habitats and food sources to lots of other animals. Recycling, reusing and reducing waste to try and avoid landfill and reduce how much actual waste goes to landfill. Reducing deforestation and the carbon dioxide emissions by governments. This is normally done through legal arrangements and getting them to sign agreements to try and reduce their impact on the environment. Seed banks can store plant genes as a way of preventing plant extinction. This also serves as a genetic diversity resource and preserves it, and these seeds can be used to replant plants in the wild. If their habitat has been recovered, then you can use these seeds to kind of repopulate areas as well. Humans have been breeding organisms for certain desired characteristics for thousands of years, especially in crop plants, livestock, and domesticated animals. The start of the process is that you would select two parents that have the desired characteristics. You would then breed these together. Then select individuals again from the offspring with the desired characteristics and breed these together. So select, breed, and then it's just about repeating that over many generations until all of the offspring, or you have many organisms that have the desired characteristics that you want. We need to know some examples of the different desired characteristics that humans have bred things for in both animals and plants. So in animals, they have been bred for more meat, milk, egg or wool yield, if we're thinking about livestock animals like cows, sheep, pigs and chickens. Also a gentle nature in domestic dogs. So our dogs are our kind of pets and they have this kind of gentle, calm, loving nature that we have bred into them. In plants, it's disease resistance in food crops and large or unusual or heavily scented flowers. So there's loads of different flowers grown for aesthetic reasons because they look nice like orchids and they have been selectively bred. There are some issues with selective breeding. Inbreeding reduces genetic variation and makes organisms more likely to suffer with recessive genetic disorders. It's also more likely that they will be susceptible to new pathogens and other diseases because, again, they have very little chance of having resistance because they have low genetic variation. Genetic engineering involves cutting a gene out of a chromosome and transferring it to the cells of a different organism to give it a desired characteristic. Some examples are crop plants that have been modified to make them resistant to pests, diseases, herbicides, or for a greater yield. They can also have improved nutritional value, like golden rice. Bacteria have been modified to produce useful substances, for example, human insulin, to treat type 1 diabetes. We need to be able to evaluate the benefits and disadvantages of genetically modified crops. There is a concern that any genes in plants could spread to wild populations. That means that they could also affect food chains by killing insects, for example, or biodiversity. There's a concern that they could cause allergic reactions in humans if eaten. And people are concerned that there has been no long-term studies on the effects of eating genetically modified crops. There are also some ethical concerns around genetic modification. Some people believe it's not natural to interfere with genomes, and there are concerns of use over this technology in humans, which is currently illegal. We need to be able to evaluate the risks and benefits of genetic engineering. One of the benefits of genetic engineering is that it's faster than selective breeding to produce organisms that have desired characteristics. Herbicide-resistant crops will reduce the need to use herbicides, which reduce biodiversity. The Bt toxin from bacteria can be inserted into plants in order to reduce the need for pesticides, because when insects eat them, the toxin will make them be ill or sick or kill them, and so they won't continue to eat the plant. Crops have their nutritional value increased. An example is golden rice, which has been genetically modified to produce more vitamin A. And sterile insects can be produced to reduce the spread of vector-borne diseases. Some risks and disadvantages. People believe that it's not ethical to interfere with nature in this way. Toxins in pollen could harm insects, specifically pollinators, and toxins in plants could enter humans and cause harm. New genes in food crops could cause allergic reactions in humans. And GM crop seed is expensive, and some developing countries may not be able to afford it, and so some people also believe that this is unethical because it is not fair that they do not get the benefits of the genetically modified crops. Health is a state of physical and mental well-being. Causes of ill health include disease, which can either be communicable disease or non-communicable disease. Communicable diseases are diseases that can be passed from one person to another, and they are caused by a pathogen. Non-communicable diseases cannot be passed from one person to another. Diet, because this can lead to certain non-communicable diseases. And stress, which can be caused by social or lifestyle factors. Different types of diseases might interact with each other, and you need to know how. Viruses can infect cells and cause cancer. For example, HPV can cause cervical cancer. Immune reactions to infections with a pathogen can trigger allergic reactions like skin rashes and asthma. So asthma is a form of allergy. Severe physical ill health caused by disease can lead to depression and other mental illnesses. Communicable diseases can spread from one organism to another because they are caused by pathogens that can be transferred. Pathogens are microorganisms that cause disease. One example of a pathogen is a virus. These are small, acellular, which means they are not made of cells. They live inside cells and replicate, causing damage to the cell when they burst out. They can reproduce rapidly inside humans. Another example of a pathogen is bacteria. These are prokaryotic cells that can reproduce rapidly inside humans. Bacterial cells release toxins which cause damage to cells inside the body, which is what gives you the symptoms and makes you feel sick. Another example of a pathogen is fungi. These can grow on the surface of leaves or skin, and they feed off living tissue. And lastly, protists. These are single-celled and often parasitic organisms that live inside and replicate inside host organisms, which cause damage. Communicable diseases can be spread through the air, through direct contact, or through vectors. An example of transmission by air is known as droplet infection. This is where we inhale pathogens in particles from coughs and sneezes. There are two forms of direct contact, either by consuming contaminated food or water that contains the pathogen, or by touching or having contact with infected tissue, for example, blood. An example of contaminated food is Salmonella, which is a bacteria that causes food poisoning. And an example of direct contact through infected tissue is HIV, which can be transmitted through bodily fluids, including blood. An example of vector transmission is malaria. So this is caused by a protist pathogen which is transmitted by a mosquito vector when it bites humans. There are different ways of identifying communicable diseases in order to help them prevent the spread. Antigens are the proteins on the surface of pathogens. You can see I've circled one here on this virus. These can be tested to see if they are present in an organism, because if you have the antigens for a pathogen, then it is likely you're infected with that pathogen. You can visually identify the pathogen either through symptoms like a characteristic rash or through identification using a microscope. DNA testing can be used. Some pathogens, for example viruses, inject their DNA into the host cells. This means that by taking a sample, we can test whether or not there is pathogen DNA present, and then this can show whether you've been infected by that pathogen or not. There are three examples of viral diseases that we have to know. The first one is measles. The symptoms of measles are kind of a red rash, so spots on the skin, and a high temperature or fever. It's spread through droplet infection because it also causes a cough, and it can cause blindness and even death, especially in children. The only way to prevent the spread of viruses like measles is through vaccinating young children. The next example is HIV. This is a virus that infects and kills white blood cells. It weakens the immune system and eventually leads to AIDS, which is where a person has a weakened immune system and will get secondary infections. This includes things like TB, which infects the lungs and can cause death. This is transmitted through sexual contact or bodily fluid contact. So one way of preventing the spread is by using condoms as a barrier method of contraception. TB is a bacterial infection, so often these secondary infections will be caused by other pathogens, and the weakened immune system just cannot fight them off, and so they're more dangerous than they would be if you did not have AIDS. The tobacco mosaic virus is an example of a plant viral disease. It infects plants and it destroys their chloroplasts in the leaves. This creates a mosaic pattern of discoloration on the leaves and reduces the plant growth due to lack of photosynthesis. You can prevent the spread of the tobacco mosaic virus by burning the whole plant and disinfecting any tools or hands that have touched the plant, and it can be spread by insect vectors such as aphids, so killing those can also help prevent the spread. There are two bacterial disease examples we need to know. The first is gonorrhea, which is a sexually transmitted infection, so we can prevent the spread with condoms, similar to HIV. The symptoms caused are discharge from the genitals and pain when urinating, and it can also cause a fever. Salmonella is the second example. This causes food poisoning that we have already mentioned, as it's spread from eating contaminated food, for example, raw chicken. The symptoms are sickness, fever, and diarrhea. We can prevent the spread by washing our hands before and after touching raw meat, and also cooking food to high enough temperatures to kill the bacteria. In the UK, chickens are also vaccinated against salmonella to try and reduce the spread and make sure it's not present in eggs as well. In both of these examples, the symptoms are caused by toxins released from the bacteria. Treatment for any bacterial infection involves antibiotics. These are drugs that kill bacteria in the body or stop them from replicating. Not all antibiotics will be able to kill all bacteria. We now have an example that there are many resistant strains of gonorrhea in the UK specifically, which cannot be treated with most antibiotics. Another example of bacterial disease, this time in plants, is called crown gall disease. It's caused by the genus of bacteria known as Agrobacterium. It causes the growth of a gall, which is like a cancer, so it causes these large tumours to form at the site of infection. The tumours can normally be seen on roots or stems. It's quite a risk because it can infect many different plant species. It is not specific. The Agrobacterium transfers its DNA into the plant cells. The DNA can be used to therefore diagnose through testing whether a plant is infected with Agrobacterium. Another bacterial disease is tuberculosis. This is spread by droplet infection. It causes lung damage, and the symptoms include tiredness and fatigue, a serious cough that can lead to coughing up blood, and fever and weight loss. A vaccination programme with the vaccine known as the BCG has reduced cases dramatically in the UK. People with reduced immune systems are more likely to catch it and suffer from TB. A fungal disease that infects plants is barley powdery mildew. It is a fungus that infects grass plants, not just barley. It causes white, fluffy growths on the leaves, as you can see here. The plant eventually cannot make any more chlorophyll, so the plant cannot absorb light for photosynthesis and will die. It's spread by spores on the wind. Spores are the reproductive cells of the fungus. You can treat it with fungicide in order to kill the pathogen, and infected leaves and tissue should be removed and burned in order to kill the spores. You can also move plants away from each other while they're being treated with fungicide and the tissue being removed, and then when they are healthy again, you can put them back close to other plants. An animal example of a fungal disease is athlete's foot. It's caused by a fungus that usually grows between people's toes. Symptoms include a red, itchy rash and flaky or white cracked skin. It's transmitted by direct contact with infected skin or wet surfaces specifically that have been contaminated, for example in pools or gyms. Treatments for it include using an antifungal cream, which will kill the pathogen, but also disinfecting surfaces, and that can help to prevent the spread. Blood is a tissue which transports substances around the body and plays a role in the immune system. The majority of blood is made up of blood plasma, which is the liquid that blood cells are suspended in. It's mostly water, but it contains all the dissolved substances that are being transported around in the blood. Things like glucose and other things like antibodies, for example, and other dissolved substances from the digestive system, like the amino acids and things that need to travel to and from your cells. The rest of the blood is made up of 3 different types of blood cells that are suspended in this liquid. They're red blood cells, white blood cells, and platelets. Platelets are involved in clotting open wounds to try and prevent infection or to prevent excess bleeding. They can do this because they have surface proteins that allow them to bind together and bind to open wounds, and they also secrete proteins that help form the clot. Red blood cells have many special adaptations to allow them to be efficient at transporting oxygen. They have no nucleus. They contain lots of hemoglobin protein instead, so having no nucleus makes more space for this. They have a biconcave shape, so like a donut but with no hole all the way through. This gives them a large surface area for diffusion of oxygen. They're flexible, so they can fit through narrow capillaries. Hemoglobin is able to reversibly bind to oxygen, so it can transport it from the lungs to the cells. This means it can bind to the hemoglobin and then it's able to release it when it gets to the cells. There are two types of white blood cells: phagocytes, these engulf pathogens, and lymphocytes. These produce antibodies or antitoxins. Both of these defend the body from pathogens and prevent you from getting sick, so they play a part in the immune system. Blood can be donated to provide useful substances or blood products to treat patients with a transfusion. These can include red blood cells, platelets, plasma, and antibodies. These are separated from the blood donation itself, and then the patients are given exactly what they need. Before it can be given to patients, blood needs to be screened. Often white blood cells are removed to try and reduce infection risk. They're screened for pathogens like HIV. The blood group of the donor is checked because it must be a match with the person who's receiving the transfusion. And there's normally a check for the presence of certain antibodies. Blood can be tested as well as looking at it under the microscope to identify the cells, and you may have done this and seen an image like this. We can recognise red blood cells, the white blood cells, the lymphocytes and the phagocytes, and then platelets. Human defence systems. Humans have some non-specific defences that try to prevent pathogens from entering the body. The nose contains hairs and mucus to try and trap pathogens. The trachea and bronchi in the airways contain cilia on the cells to move mucus, which contains trapped pathogens, to the nose and mouth to be removed from the body, or to be swallowed. The stomach contains hydrochloric acid to kill bacteria in food or swallowed mucus. And the skin acts as a physical barrier to prevent the entry of pathogens everywhere else. Specific defences work inside the body in case pathogens do enter, and they're normally carried out by white blood cells. Phagocytes detect and engulf pathogens and digest them with enzymes. Lymphocytes are triggered to divide by mitosis when they detect a foreign antigen. They will form plasma cells, which release antibodies that are specific to the antigen on that pathogen, and they can also produce antitoxins that can bind and neutralise toxins released from bacteria. Vaccines contain a dead or weakened form of a pathogen, so they will not infect the person, but the antigens on the pathogen will bind and trigger white blood cells to produce antibodies that can bind to the specific antigen on the pathogen. So you can see I've got my pathogen, my antigen, which is kind of the spiky protein on the outside, and my purple antibodies, which can bind to that antigen. The white blood cells will form memory B cells that will remember the antigen on the pathogen and will produce more antibodies faster next time they are reinfected in order to prevent illness. This makes people immune to that pathogen. So the first time you are infected with pathogen or an antigen from a vaccine, we go through the primary immune response to make antibodies for the first time. So not many antibodies are made, and it happens slowly. The second time you get infected with the same antigen, so if you inhale it naturally, for example, through droplet infection, we have what's called the secondary immune response. So because you have memory cells, they produce the antibodies faster and there's more of them, and so we get that bigger peak in the graph that we can see here for the secondary immune response. This prevents you from getting sick and gets rid of the pathogen from the body before it has a chance to make you ill and before you can pass it on to other people. Vaccinating large numbers of a population can prevent the spread of a disease, because making people immune means they cannot be infected with the pathogen and so they therefore cannot pass it on to other people. This is known as herd immunity. Examples of painkillers include ibuprofen, paracetamol, morphine, and codeine. Painkillers are taken to treat symptoms of diseases such as pain, inflammation, and to lower temperatures caused by fever. Painkillers do not treat the cause of the disease because they do not affect pathogens. Examples of antibiotics include penicillin, amoxicillin, and tetracycline. Antibiotics are not antiseptics or antivirals. Antiseptics kill bacteria outside of the body, for example, on surfaces. Antibiotics are drugs that kill bacteria. They treat the cause of the disease by killing the pathogen. Antibiotics cannot and do not work on viruses because these replicate inside cells and bacteria live outside of cells. So antibiotics cannot get into or work inside cells. Antivirals can be used to stop viruses replicating. So they are drugs used to help treat viruses that are different from antibiotics. Antibiotic resistance. Some bacteria in a population have the ability to survive treatment with an antibiotic. These will survive and reproduce, passing on the genes that allow them to survive to their offspring. Over time, we get an increase in frequency of the resistant bacteria in a population until we have a whole population of bacteria that cannot be killed by that antibiotic. This is an example of natural selection and therefore evolution of bacteria. We need to be able to suggest ways to reduce antibiotic resistance. We can reduce prescriptions of antibiotics for viral infections, so prevent doctors from giving out antibiotics unnecessarily. Making sure that patients take the full course of antibiotics every time, so they take all of the antibiotics they're given, even if they start to feel better. And reduce the use of antibiotics in farm animals. Culturing microorganisms. Bacteria divide really rapidly by a process called binary fission. They can split into two as fast as every 20 minutes. You can calculate this by using the following formula. You take the number of bacteria that you start with, and then you multiply by 2 to the power of the number of divisions that have occurred. Bacteria can be grown in a liquid broth culture or on nutrient agar, which is like a gel, plates. In order for them to grow successfully, they must have enough nutrients and oxygen, and also be warm, so be at the right temperature, and there should be no contaminants. So there should be nothing else, no other microorganisms that can grow there that could contaminate the sample. So in order to prevent that, the first thing we should do before using these is to sterilize the broth, so the liquid medium or the agar plate, before you put any bacteria on it. So to produce our uncontaminated plates, we need to use the aseptic technique that you've seen in the video, where you flame all your instruments that are metal or glass, flame the neck of any open bottles. Don't place anything down onto the bench, you should be holding it at all times. Once you need to put it down or you're finished, you can put it into disinfectant. You should disinfect surfaces and your hands before and after you carry out the practical, and you should always work near an open flame, which produces a hot airflow and carries any contaminants away from where you're working on the surface. After you've made your plates, for example, if we're going to look at antibiotic resistance and have put some discs on top of some bacteria on a plate, you need to incubate them at 25 degrees so that we don't get any pathogenic bacteria growing. We need to loosely tape the lids shut to have a lid sealed on top, but not completely sealed, so the bacteria can still have oxygen in order to be able to grow. The plate should also be placed upside down so that no condensation can fall on the bacteria. Once we've left our plates to grow for a few days, we should observe the colony growth and look for any clear zones around our antibiotic discs. You can then measure the diameter in 2 places, normally at right angles to each other, and then use a mean of these two values and pi r squared formula to calculate the area of the clear zone around the disk. Larger clear zones around the disk will mean that more bacteria have been killed, and so the antibiotic is better or stronger. Originally, drugs were extracted from plants and microorganisms. There are 3 examples we have to know. One is aspirin, which is used as a painkiller, and this was extracted from willow tree bark. Digitalis, this is a heart medication that's extracted from the foxglove plant. And penicillin, which is an antibiotic, which comes from the penicillium mold, which was discovered by Alexander Fleming. New synthetic drugs made in the pharmaceutical industry can still have plant extracts as their base, but they must be tested for 3 things: toxicity, efficacy, and dosage. Toxicity is referring to whether or not it has serious side effects or is toxic to cells. Efficacy is whether or not the drug works to act in the way that we want it to. And dosage is about how much drug needs to be taken for it to work whilst not having too many side effects. We need to know the 3 stages of drug trials, what happens in each, and what they are testing for. The first stage is carried out in the lab, and it is known as preclinical testing, where we test the drugs on cells, tissues, and animals. At this stage, drugs are being tested for toxicity and efficacy. In the second stage, we move on to clinical trials, which involve humans. In clinical phase 1, we use healthy volunteers, and low doses are used to check for side effects. In clinical trial phase 2, we would use patients who are sick with whatever the drug is supposed to help with, and then we are testing for efficacy and dosage. Drugs cannot proceed through these drug trial stages unless they pass the checkpoints. So only if a drug is not toxic and found to work in the preclinical stages will it pass onto the clinical trials with humans, and only if the drug passes the clinical phase 1, where it's shown to not have serious side effects in humans, would it be given to patients, because it is unfair to give or risk patients' health or making them worse by giving them a drug that could cause serious side effects or that does not work. Clinical trials should be at least blind clinical trials. This is where we use a placebo or a fake drug as a comparison to show that the real drug is actually having the effect. In blind clinical trials, only the patients do not know if they are getting the real drug or the placebo. In double-blind clinical trials, neither the patients or the doctors know who is getting the placebo. This is the best standard of clinical trial. Double-blind clinical trials are important because they reduce the potential bias from the doctors. It reduces the psychological effects, for example, the placebo effect, where patients may just report feeling better because they think they are getting the real drug. And it can show that the real drug is having an effect when compared to the placebo. So we know for sure it is the drug that is working and actually having a measurable effect on the illness. Non-communicable diseases cannot spread between organisms. They're not caused by pathogens. They are usually long-term and get worse slowly. Examples include asthma, diabetes, and heart disease. Risk factors can increase the chance of getting these diseases. Non-communicable diseases can be caused by genes or genetic factors. These are out of a person's control. They could be caused by a mutation, or they could be inherited from their parents. The following are all risk factors related to lifestyle and often choices. Alcohol has been linked to effect in unborn babies' brain development. Excessive alcohol consumption has also been linked to liver disease and brain damage. A high-fat diet can lead to obesity. Obesity is a risk factor for type 2 diabetes, and a high-fat diet can also lead to high cholesterol, which is a risk factor for heart disease. Smoking whilst pregnant can reduce the birth weight of unborn babies. Smoking has been directly linked to causing lung cancer and lung diseases, and it also increases the chance of heart disease. Ionizing radiation, this is a link to your physics specification. So a carcinogen that damages DNA in cells, so it's an example of that. Exposure to ionizing radiation has been linked to cancer. For example, UV ray exposure has been linked to an increase in skin cancer. So you've got this idea that ionizing radiation will damage DNA, and then that can lead to it causing cancer, which we're going to look at next. But most diseases will be caused by a mixture of different factors coming together. It's not always just one of these. Coronary heart disease. The coronary arteries are blood vessels which transport blood to the heart muscle itself. If these coronary arteries get blocked with a fatty buildup called plaque, then blood flow is reduced, so less oxygen and glucose can reach the heart muscle. Without enough oxygen and glucose, the heart muscle cannot respire, so it stops contracting. This can cause a heart attack. Some risk factors for coronary heart disease, that means they increase the risk or the chance of you getting coronary heart disease, are poor diet and lack of exercise. This is because they can lead to other conditions such as high blood pressure, high cholesterol, and obesity, which are all medical risk factors for coronary heart disease. We need to be able to describe some treatments for coronary heart disease. Statins are drugs that can be taken to lower cholesterol. These are a preventative measure, so they cannot reduce any buildup that is already there, but they can reduce future buildup of plaque or fat in the coronary arteries in the future. Stents are a metal mesh-like cage which is used to widen arteries that have been narrowed by this fatty buildup. This can help to increase blood flow. With serious coronary heart disease, it can cause heart failure. If heart failure has been caused, then the options for treatment are a heart transplant or a heart bypass, or an artificial heart, which can be used to keep someone alive until a transplant becomes available. You need to be able to discuss the advantages and disadvantages of these different types of treatments for coronary heart disease. Lifestyle changes, including a healthier diet, more exercise, reducing alcohol intake and stopping smoking, can also help to dramatically reduce the risk of coronary heart disease. It can also be part of treatment. If being given statins, for example, you can be encouraged to make these lifestyle changes to help reduce your risk further. Cancer is a non-communicable disease where uncontrollable cell division causes tumours to form. There is some damage to the cell, normally to the DNA. This alters the way the cell division process occurs. This leads to uncontrolled or unlimited cell division, which then forms a tumour. Some tumours can be classed as benign. These are slow growing and non-cancerous because they are contained in a membrane and do not spread to other parts of the body or other tissues. Malignant tumours, though, are cancerous. They are fast growing and they can break off and spread to form secondary tumours in other parts of the body or other tissues by travelling through the bloodstream. You need to be able to define cancer as uncontrollable cell division and know about the two different types of tumours. Risk factors for cancer. Smoking introduces carcinogenic chemicals into the body through the tar that's in the cigarettes, and it can cause lung cancer, but others as well. Exposure to UV radiation, which we said is a form of ionizing radiation, has been linked to skin cancer. Obesity is a risk factor because we know that fat cells can signal other cells to divide. Viral infection, we've talked about the example with HPV that can cause cervical cancer. Some cancers can be caused by genetic factors, so either mutations in DNA or inherited genes that can increase your chance of getting a certain cancer. For example, the BRCA gene, which is known to increase your chance of getting breast cancer. Therapeutic cloning can be used to produce embryonic stem cells that are genetically identical to the patient. These can then be used to grow new cells or replace damaged ones. The hope is that one day they can also be used to grow whole organs, because this can help to reduce the issues with rejection around donor organs, and it can also help reduce the issue with the fact that we have a low amount of donors, and it would mean that the patient would not need to take immunosuppressant drugs for the rest of their life, because the organs they would be having would be made of their own stem cells, so it would be genetically the same as them. There are many different uses of stem cells. We can use them to produce new brain cells to treat Parkinson's disease, rebuild bone and cartilage after damage, make replacement heart valves in order to treat heart disease, or repair damaged immune systems. This is normally done with a bone marrow transplant, as bone marrow contains the stem cells that create new blood cells. Evaluating the use of stem cells can be difficult. It very much depends on the stem cells you're talking about, whether it be adult, embryonic, or stem cells produced for therapeutic cloning. There's lots of factors to consider, and there is often no right or wrong answer. As long as you can answer exam questions with an evaluating, balanced argument, then that is fine. Evaluating the use of stem cells means that we have to look at the clinical, social, and ethical issues. Clinical issues are that there are no guarantee of success. Not every stem cell treatment will work. We need stem cell donors, and accessing those can sometimes be difficult. Also, collection and storage of embryonic stem cells can sometimes be difficult due to lack of space or the cost. Umbilical cord blood can be saved from birth, but this needs to be planned in advance so that it can be saved and frozen and kept for that person for their life in case they are in need of those stem cells. Stem cells have been known to mutate and could cause cancer when placed in the body. There's also a risk of transferring a virus if you're transferring stem cells from other people. Social issues. We need to educate the public about the limitations of stem cells, what they can and can't do, because some people might believe that they are a cure-all treatment. We need to listen to people's objections and see if these outweigh the benefits of using stem cells. Private clinics can charge people a lot of money for experimental stem cell treatments that haven't been through the peer review process, so they may not work, leaving people very disappointed. Ethical issues. People have issues with the use of unused IVF embryos, also issues around the creation of embryos for therapeutic cloning that could then be destroyed. There is a concern that embryos could become a commodity, so they could be something that is bought and sold and seem to be really valued and important, but not as a human life, but as something that can be used as a medical treatment. Issues around consent, because it's about deciding when an embryo becomes a person, and obviously they don't have the right to consent to be used for stem cell research. Unlike adult stem cells, which obviously can be donated from people who have consented. Gene therapy involves replacing a faulty allele that causes a genetic disease with a functioning one. An example is cystic fibrosis. A dominant functioning allele of the CFTR gene can be inserted into lung cells to try and reduce the symptoms caused by the faulty alleles in those cells normally. The method involves identifying the gene involved in the disorder, for example the CFTR gene in cystic fibrosis. fibrosis. Cutting out a functioning allele using enzymes, this could be from a sample of someone who has not got cystic fibrosis. Many copies of that allele must then be made, and then these alleles must somehow be transferred into the cells of the person with the disorder. Many of the issues lie with the last step in this process. It's very difficult, and there's different techniques to try and get all of the alleles into all of the cells, but they may not actually make it into every cell. These treatments are not permanent because transformed cells are replaced regularly in the body with cells that have the incorrect gene again. The alleles may also insert incorrectly into the chromosome and might not work properly. There are also ethical issues around gene therapy. It's illegal to do in gametes or sex cells because this would then be passed on to the offspring. So the new gene or the added gene could be passed on to the offspring, and there could be a chance of mutation or other genetic issues that we don't know yet. This means that if we're not targeting sex cells, then gene therapy at the moment is only carried out on body cells, which means it's not a cure because it's only used in body cells. As we said, it's temporary, they can be replaced, and as sex cells are not being altered, they can still pass on their disorder to their offspring. People also do not agree that human DNA should be changed. The Human Genome Project was completed with global collaboration to sequence the 3 billion pairs of bases in the human genome. And since we've been able to do that and understand and start picking apart what actually this means, we've been able to improve and hopefully increase in the future our ability to treat humans using the knowledge that we have of the genome. One area specifically is in genetic disorders. So being able to identify genes that actually cause genetic disorders or increase the risk of other diseases allows people to make informed decisions. For example, breast cancer genes have been identified that if you have certain versions of these genes, they can increase your risk of contracting breast cancer later on in life. People can be screened for these, and then if told that they have these specific mutations, a patient can decide whether or not to have preventative surgery to remove breast tissue to dramatically reduce their chance of getting breast cancer. For other genetic disorders like Huntington's disease, gene testing can tell people if they are a carrier or if they're likely to pass on a genetic disorder to their offspring, so they can choose and make informed decisions when deciding whether or not to have children. Targeted therapies and medicine. Gene mutations can be identified that cause disease. For example, forms of cancer can be caused by specific mutations. These can then be used to make targeted drugs to the individual so that they're more likely to work for that person and their disease. For example, a specific form of cancer was discovered that is caused by a specific mutation in a specific gene that results in a specific protein. A monoclonal antibody treatment was able to be made that can bind to and deliver drugs to this targeted protein, and therefore everyone who can be screened and tested for this type of cancer can then be given this treatment, and there's a high chance that it will work. In the future, analysing the human genome can help us to improve more types of gene therapy and also more personalised medicine.
Ouch! This is why in some videos I'll explain scratches.